everybody. Today we're going to be talking about Krebs cycle regulation. So how the cell decides how much of the Krebs cycle it's going to do, if it's going to do the Krebs cycle at all. It's important to make enough energy using the Krebs cycle in the cell, but you don't need to make too much. You just need to make the amount that you need. And then if you have enough energy, you can be doing other things other than just continuing to make excess energy. We've touched on this already when we're thinking about pushing glycolysis up into some of those shunts, like the pentose phosphate pathway or into glycogenesis. And we've talked a little bit about how we can get into fatty acid synthesis from the Krebs cycle directly. There's some amino acid interaction with the Krebs cycle, and all of these things are going to be dependent on various feedback mechanisms that will determine which way reactions are going to go, if we're going to continue on with the Krebs cycle or if we're going to push it into one of these other directions. So conditions inside the cell or presence of conditions inside the cell, presence of various enzymes, determine whether we continue with the Krebs cycle or push the reactions into other directions. So everything is very much tied together, whether we're thinking about carbohydrate metabolism and glycolysis, glycogenesis, glycolysis, I just said that. So all of those things having to do with glucose, or whether we're talking about breaking down lipids or making lipids, or breaking down proteins or making proteins. A lot of these things are really, really tied together, and they're branching off either from glycolysis or from the Krebs cycle itself. So that's what we're going to look at with this video. Alright, so here's our Krebs cycle. Again, we don't need to memorize the Krebs cycle, but we do need to point out some parts of the Krebs cycle. It should be looking pretty recognizable. We've seen this chart quite a few times here. We see pyruvate. That was from glycolysis that happened before. So glycolysis happens, we end up with pyruvate. We use pyruvate dehydrogenase. That's this enzyme right here, which I know you can't see that on here, so I'll write that in. Pyruvate dehydrogenase. That's an enzyme that we just keep seeing, so it should be starting to look familiar to you. That's what takes our pyruvate and turns it into acetyl-CoA, and acetyl-CoA can enter the Krebs cycle, interacting with oxaloacetate. I'll write that in there to make sure you can see that, oxaloacetate, which is another one that we keep seeing. Oxaloacetate, we're going to see this over and over again. It's involved in a lot of different reactions. And then we're in the Krebs cycle starting at citrate. Other things that we want to make note of, other than just our pure entering into the Krebs cycle, running through the Krebs cycle, getting some energy out of it, and getting NADH and FADH2 for the electron transport chain. We know all that stuff already, but we're looking more at where we can branch out or where we can feed in to the Krebs cycle, other than just acetyl-CoA. Acetyl-CoA can come from other sources other than just glycolysis. It can come from breaking down of fatty acids. It can come from breaking down of certain amino acids. So we get this acetyl-CoA from multiple sources, and it can go through this whole energy creating cycle, or if we have certain conditions in the cell or certain enzymes present, we can have little side branches. One place to notice, especially on the Krebs cycle here, then would be isocitrate. And here's this enzyme, little tiny writing. I'll write it out bigger so you can see it, but it's almost guessable what the name of this enzyme is going to be. It's isocitrate dehydrogenase. This is a branching off point for some fatty acid synthesis. We also need to look at alpha ketoglutarate. That's something else that we have seen before. And then as we get into over here, succinyl-CoA and fumarate. These molecules are all going to have something to do with amino acids coming into the Krebs cycle or going out of the Krebs cycle. So there's all these places where we can have essentially shunts off of the Krebs cycle. Not just the shunts that we've already seen off of glycolysis, but also these little side branches coming off of or going into the Krebs cycle itself. Now here, this bullet point here, it's saying whatever pathway you're using, whether it's one with fatty acids, whether it's continuing the Krebs cycle, or using amino acids, or creating amino acids, or bringing them down, kind of incorporating them into or relating them to the Krebs cycle, these enzymes that are part of each transformation along the way are going to be part of how we decide which way the reaction is going to go. So if you have a lot of, say, pyruvate dehydrogenase, that means you're going to make a lot of acetyl-CoA and feed into the beginning of a Krebs cycle. And maybe you do that, and then you also happen to, I don't know why I wrote that way, it should say isocitrate dehydrogenase, sorry, isocitrate. My mouth moves faster than my pen. So isocitrate dehydrogenase, it changes isocitrate into oxalosuccinate. If we have a lot of this enzyme, then we're going to go off in that direction into another pathway here, a fatty acid synthesis pathway. So all of these different enzyme amounts are going to determine which way we go. And if we go off into the fatty acid synthesis pathway, then we're not going to be doing this so much. This is all going to slow down. So the whole rest of the Krebs cycle will be effectively inhibited because we're taking our products and sending them off to a different place. So we're using them instead of in the Krebs cycle, we're using them for fatty acid synthesis. So it'll affect everything on down the line. So remember, if anything proximal to that, so anything before that, wouldn't be affected by a high amount of isocitrate dehydrogenase, but all the reactions here distal to isocitrate dehydrogenase would be affected if we are going to be sending those enzymes off to a different place. Okay, so this is just kind of went over what we just talked about, but just again to just notice pyruvate dehydrogenase, we're going to focus on that and how it, gives you acetyl-CoA to begin the whole process. Our isocitrate dehydrogenase, which is going to help to catalyze this part of the reaction, and this is also where we branch off into, say, fatty acid synthesis. It's having you take note of alpha ketoglutarate, like we mentioned before in the last slide. Remember, alpha ketoglutarate, this has an amino acid on it, so there's some amino acid synthesis here. Or we'll just say amino acid metabolism, because amino acids can go in either direction, either going away from the Krebs cycle, going into the Krebs cycle. We'll also look at the succinyl-CoA, and in desuccinate and fumarate, there's a lot of amino acid stuff going on in this whole area. Okay. And then oxaloacetate, which should sound kind of familiar to you? We've seen oxaloacetate. We've talked in detail about oxaloacetate with the malate aspartate shuttle. Remember, oxaloacetate was involved on both sides of the mitochondrial membrane, whether inside the cell or inside the mitochondria or in the cytoplasm, outside of the mitochondria, helping to accept or get rid of amino acids so that we can shuttle aspartate and glutamate across the mitochondrial membrane. So we've seen oxaloacetate in action before. It's part of the Krebs cycle, but it's also something that can be created directly from pyruvate, and we can also reverse oxaloacetate and end up in gluconeogenesis. So going back into, instead of breaking down carbs like we do with glycolysis, we can be creating carbohydrates through gluconeogenesis just by using the oxaloacetate molecule. So everything is so intertwined here, and all kind of converges on the Krebs cycle and glycolysis. So we can really focus on each one specifically. So for Unit 2, we're going to be looking a lot at fatty acid synthesis and how we use fatty acids in the Krebs cycle, how we break them down, things like that. As we get into Unit 3, we'll be looking more at amino acid metabolism and how we create amino acids, how we break down amino acids, things like that. In Unit 1, we already looked at a lot of what we do with carbohydrates, how we break it down to get energy, how we do glycogenesis to make glycogen to store excess glucose, and also a little bit of how we do gluconeogenesis. So just kind of finishing up with our carbohydrates a little bit here so we can move on to these other pathways. Okay, so here we have a different schematic of the Krebs cycle. It doesn't show glycolysis on here, but it shows pyruvate. So glycolysis goes into creating pyruvate. And then we see acetyl-CoA, and we're entering into the Krebs cycle by reacting the acetyl-CoA with oxaloacetate, forming citrate, which is essentially the beginning of the Krebs cycle. And then we have some offshoots here, right? So we have, and this little arrow should be a little bit past the citrate. I would kind of put it more like that, but we can take our acetyl-CoA and feed it into fatty acid synthesis. And there's a process, and we're going to look at the process of fatty acid synthesis in a few videos from now, so as we get into the actual process of making our fatty acids. We've touched on it in our last video. We saw malonyl-CoA. We noticed or talked a little bit about where that came from and how that came out of the Krebs cycle, and then how we ended up with fatty acids. But we're going to look at that in a lot more detail in future videos. Here we go with isocitrate, and this here would be isocitrate dehydrogenase, would be the enzyme. That takes us on to the next step. Alpha ketoglutarate. The name keto acid, makes you think of proteins and an amino acid essentially. So when we look at alpha ketoglutarate, we know that we can interact that with glutamate. We saw that in the malate aspartate shuttle as well, where we are taking an amino acid and just changing it, exchanging it, transaminating from one molecule to another. And here we see another amino acid, glutamine, okay, and there's an amino acid coming off of that, or ammonia, I should say, not an amino acid. Coming off of that, so we have this exchange of amino acids going into the Krebs cycle and going out of the Krebs cycle. It can go in either direction. This is a reversible arrow here. These are all amino acids that can feed into the production of glutamate. So these are particular amino acids that are used to create the glutamate molecule, and then they can be taken off, exchanged, transaminated. Over here with succinyl-CoA going into succinate, these are amino acids that can become incorporated into the succinyl-CoA, or that can become incorporated into the Krebs cycle at this spot on the Krebs cycle reaction chain. At fumarate, we have these amino acids entering into the reaction here. And then at oxaloacetate, there's aspartate. We saw that in the malate aspartate shuttle. That can be used to, shuttle across the mitochondrial membrane. So that's an important situation here. We know that's reversible. We've seen that reversing already in that malate aspartate shuttle. But this here, asparagine, that's also an amino acid. So you can just see all these different kinds of amino acids and how they're being exchanged from one molecule to another, and it's all integrated with the Krebs cycle. That is the whole point of that. So let's use a different color so we can kind of highlight some stuff here. Just to think of fatty acid synthesis happening over on kind of the top initial part of the Krebs cycle in the beginning. So from citrate through isocitrate all the way through there, we get into fatty acid synthesis, so the shunt going off in that direction. When we start to hit alpha ketoglutarate and really going all the way over to oxaloacetate, the rest of this is going to be amino acid metabolism, whether we're going just into, so heading into the Krebs cycle over here, or going in either direction, going in or out, as we have over here at alpha ketoglutarate or over here at oxaloacetate. Another take-home message from this slide is, looking again at oxaloacetate, we have another enzyme over here called PEP, or phosphoenol pyruvate carboxylase, which is a mouthful. So PEP just sounds a lot better. It's a lot easier to call it that. And through an intermediary product, which we don't need to worry about too much, I just put it there just to show you. So this is an intermediary product. We can get glucose out of that. So at the end of the Krebs cycle, before we're even, going to start another round of the Krebs cycle by reacting acetyl-CoA with oxaloacetate, instead of doing that, we can just push that oxaloacetate, because of this enzyme, off into gluconeogenesis, so making new glucose. And we're doing that just to create glucose to use in glycolysis. So if you're low on glucose, then this enzyme, PEP, is going to be helpful to help you make new glucose out of whatever substrate. Maybe you had a fatty acid coming in to create acetyl-CoA or a ketone from either a fatty acid or from an amino acid. Both of those things can be used to make acetyl-CoA. So maybe you don't have glucose at all. Maybe you're not doing glycolysis. You're using fatty acids or proteins, which isn't desirable for the proteins to be used, but you could be using them to get your acetyl-CoA. Or, any of these other ways we have of getting these molecules created, but you don't have glucose, then you can use this over here, oxaloacetate, into glucose, and turn around and put that right through glycolysis and have a faster source of energy. So everything is just super tied together. Okay, so this is a simplified version, I should say simplified version, to little bunny ears quotes. If you just ignore all the little enzymes they got going on here, this is just showing you in a different color scheme what I was just talking about, where we have glycolysis going into the Krebs cycle. Over here we have fatty acid synthesis. here is calling it glutaminolysis. That was just, glutamine and glutarate going into and out of the Krebs cycle. So over here, amino acid, protein synthesis. So all this over here with our amino acids happening. Towards the end, we can go into gluconeogenesis, going back over to glucose. So just showing you in a different kind of way, everything all tied together. Okay, so remember that enzyme isocitrate dehydrogenase. So over here, I'll write it down again. I'll spell it out fully this time. Isocitrate dehydrogenase. And it's used to take our isocitrate and turn it into the next step, which is oxalosuccinate. This enzyme is either inhibited or stimulated by specific things. So we either have a lot of it or we have not a lot of it. And if we have a lot of it, then we will encourage this part of the Krebs cycle, so going on through the rest of the Krebs cycle. If we don't have a lot of it, then we will inhibit everything distal to that. We'll slow down this part of the Krebs cycle. So, when do you want to slow down the Krebs cycle? You want to slow down the Krebs cycle when you have a lot of energy already and you don't need to make more energy. So when you're in the fed state or the high energy state, you have a lot of NAD, you have a lot of NADH, you have a lot of ATP. That's going to slow down this enzyme, meaning slowing down the rest of the entire Krebs cycle, less energy being created, but that's fine. You got a lot of energy at this point. That allows you to go off into your fatty acid synthesis. Okay. Now, if we stimulate it, if we had the opposite situation, so we were in a low energy state, so we didn't have a lot of ATP, we didn't have much NADH, so NAD would be present, but not having a lot of NADH because we're in the low energy state, then this would not be inhibited. It would be allowed to act, and we would speed up this part of the Krebs cycle, which would enable us to make more energy. Okay, pyruvate dehydrogenase. Remember, we go from pyruvate to acetyl-CoA, and our enzyme there is pyruvate dehydrogenase. So this enzyme is necessary to go from glycolysis into acetyl-CoA, and then yada yada yada, we have the Krebs cycle and we get energy. So it's important in order to create energy. So if we stimulate pyruvate dehydrogenase, we're going to get more acetyl-CoA, we'll get more energy, we'll run the Krebs cycle more. If we have less pyruvate dehydrogenase, so if we are inhibiting the pyruvate dehydrogenase, then we're not going to make as much acetyl-CoA, and we're not going to be able to run the Krebs cycle as much, at least from glycolysis. So what stimulates it? What inhibits it? It's stimulated by this enzyme, pyruvate dehydrogenase. So that's a type of dehydrogenase phosphate. So pyruvate dehydrogenase, pyruvate dehydrogenase phosphatase. These are tongue twisters. And pyruvate dehydrogenase phosphatase we can abbreviate as PDP. So that's pyruvate dehydrogenase phosphatase. And this would be stimulated by high levels of calcium, a lot of magnesium, insulin, and CAMP. So let's think about insulin and CAMP for just 1 minute. CAMP is a stimulatory kind of second messenger. So anytime you have CAMP activated as a second messenger in a cell, it's going to be increasing the activity of that cell. And one of the ways that it can do that is by stimulating this molecule pyruvate dehydrogenase phosphatase, which causes you to make more of this, which causes you to make more of this, which causes you to make more of this, which enables you to do more of whatever it is that cell is doing. So that makes sense. Insulin also makes sense. If you think of what insulin is doing, insulin causes glucose to enter cells. So let's say you've just eaten, you're in the fed state, so you're absorbing a lot of glucose from whatever you just ate. That glucose can go into the cells because you have released insulin in that situation. In that fed state, you release insulin, the glucose goes into the cells. That is the reason for the insulin, is to get the glucose into the cells so that you can make energy. So that being tied to this pyruvate dehydrogenase phosphatase makes sense also, and that it would stimulate it. So you can see here where we have pyruvate dehydrogenase, I'm just going to call it PDP from now on so I don't keep tripping over that word. And here it's using high levels of calcium in the cell. High levels of calcium can be stimulated by other second messenger systems to stimulate a cell, so that is where that would tie into it. So, using, like, let's just say protein kinase C or phospholipase C, if you remember that from your second messengers. We don't need to go into specifics about that. You don't need to write that down or memorize that or anything, but just as an example, other second messengers, let's say, can cause a high level of calcium being available in the cytoplasm of the cell, which can then help to stimulate PDP, which, stimulates energy production, and that's why they'd be stimulatory. And here there's one, and there's two, so there's two forms of PDP. Just think of them together. They are activating or stimulating our pyruvate dehydrogenase, which will take our pyruvate, turn it into acetyl-CoA, and then we can get energy out of that. Now, to inhibit our pyruvate dehydrogenase, so this right here, we would use pyruvate dehydrogenase kinase, or PDK. So pyruvate dehydrogenase kinase. That slows down the pyruvate dehydrogenase. So anytime you have PDK active, you're going to inhibit your pyruvate dehydrogenase. You make less acetyl-CoA, you make less energy. So what causes you to make more PDK and therefore inhibit your pyruvate dehydrogenase? The fed state, essentially. So let's just put that here. Oops, except for this last one here. Fed state. For low oxygen, you're using that to run the Krebs cycle a lot when you're in the fed state, having a lot of acetyl-CoA, having a lot of NADH, having a lot of ATP. So all these things, you have a lot of energy occurring, then you would stimulate PDK, which inhibits pyruvate dehydrogenase and inhibits the Krebs cycle, therefore. Now this last point here, starvation and nutrient deprivation, that would seem, okay, that's opposite. Why is that? That doesn't make any sense. That here you are in the fed state, you have an excess of energy. Here you are literally starving, which is the exact opposite of the fed state. You're in the starvation state instead. Wouldn't you want to do this? No, is the answer to that. And the reason why, you don't have a lot of resources available. This is when, if if you think about going on like an extreme diet, when somebody goes on an extreme diet, it stops working after a while, and people will be like, oh, my metabolism slowed down because I restricted my calories too much. We've all heard somebody saying that, or heard something about that before, that if you don't eat enough, your metabolism is going to slow down. That's because of this. This is a prevention of death mechanism, where if you think about being like a cave person, and maybe it's like winter time, and there's no nuts and berries to gather on the ground, and maybe you're not good at catching animals or something, so you're just starving, and there's not gonna be food available anytime soon. If you want to conserve what you got, just slow everything down, almost like not a hibernation state exactly, but kinda. Everything's just slowed down. You're not making a lot of energy. You wouldn't feel good in this situation. You would feel sluggish and tired and just blah. But it would allow you to exist for a little longer on what few fat stores and stored energy sources that you did have if you slowed everything down. So that's why that is there. And then there's also this, arsenate and organic arsenical. That's really just, if you think about arsenic, that's a familiar word. It's an element, it's a naturally occurring element, but it's also a poison. That is permanent inhibition of pyruvate dehydrogenase. So that doesn't sound good, right? Permanent inhibition is, permanent. It's not reversible. It doesn't mean you can never make new pyruvate dehydrogenase, but what pyruvate dehydrogenase you have in that moment, if you ingest enough arsenic, you can permanently inhibit that pyruvate dehydrogenase, and it can cause massive problems if you inhibit it, say, all of it or most of it. We'll get back to the arsenic in a minute, but let's just look now briefly at why we use glucose versus lipids as an energy source. Lipids are preferable as an energy source. Lipids are more efficient. Why is that always? I'm going to just scooch this up. The light is just too bright down there. So lipids are preferable as an energy source. They're more efficient, so you get more energy for each carbon of lipid versus a carbon of carbohydrate, but they are slower. So it takes longer to get the energy out of the lipids. And part of it is how you break them down. It's a process called beta oxidation. It takes a little bit longer. There's more steps to it. It's not as fast as glycolysis. It's a more complicated molecule than glucose. Obviously, a fatty acid, it's got a lot more going on with it than a glucose molecule. So it takes longer. But you still get acetyl-CoA, and then you can run the Krebs cycle. Lipids create a lot of acetyl-CoA when they're broken down. You get a lot of NADH and a lot of ATP. They inhibit your pyruvate dehydrogenase, so you do less of turning pyruvate into acetyl-CoA. So that essentially turns off the glycolysis pathway, enabling you to go into something like glycogenesis instead, to where you can store whatever excess glucose you have, or into the pentose phosphate pathway. So just going kind of backwards up into one of the shunts off of glycolysis. So if you have lipids available, you'll use them, unless you have a need for really quick energy. So if you were to just start doing, burpees, say, I'm just trying to think of like the most high intense energy exercise I can think of right now, like a burpee is just gonna use up your energy super, super fast. You can't use lipids for that. That would just be too slow. You would just run out and turn into a puddle on the floor. So you would then shift into using glucose because you need it faster. Okay, so arsenic / arsenate slash arsenicals. They're all something having arsenic in them. The abbreviation for arsenic is AS, so that's the chemical symbol for arsenic. And what arsenic does is it interferes with glycolysis. So I've written on the back here just a comparison of glycolysis without arsenic, so just normal glycolysis. So I didn't put all the steps here, but just like glycolysis, a bunch of stuff happens. We get to glyceraldehyde 3-phosphate, so getting kind of like towards the middle end of glycolysis. We react an NAD plus with a phosphate. We end up with an NADH and hydrogen, and then we end up with our 1,3-biphosphoglycerate. So up here we had a molecule with one phosphate stuck to the 3-carbon, so a phosphate on the 3-carbon of glyceraldehyde 3-phosphate. We added a phosphate, we put it on here, and now we have a 2-phosphate molecule. So biphosphoglycerate stuck to the, still on the 3-carbon, and then we put another phosphate on the 1-carbon, and then we go through the rest of glycolysis. We get some ATP, we get our pyruvate into acetyl-CoA, etc. But over here, if we have arsenic, so remember AS is arsenic, it reacts just like phosphate does in this situation. So we have our glycolysis, blah, blah, blah. We get our glyceraldehyde 3-phosphate. Everything's fine and dandy. But here we're doing NAD+, okay? And instead of phosphate, oh, there's an arsenic being put onto here. We still get NADH plus the hydrogen, So that still comes out okay, but what we did, instead of incorporating this phosphate into the molecule on the 1-carbon, we incorporated the arsenic into our molecule on the 1-carbon. We still have our phosphate on the 3-carbon. We didn't take that away, but that 1-carbon doesn't have a phosphate on it, it has an arsenic. So we have 1-arseno-3-phosphoglycerate instead of 1,3-biphosphoglycerate. So we can't go blah, 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 getting ATP and pyruvate. We're done now. This isn't going to go anywhere. So what you've essentially done with the arsenic, not only have you inhibited your pyruvate dehydrogenase, so whatever pyruvate you're making, because, you know, unless you're completely flooded with arsenic, you would still have some phosphate and be able to make pyruvate. It's just that a lot would be contaminated, depending on how much you ingested, a lot would be contaminated as this. But you'd still be doing this, you know, both at the same time. So this arsenic is inhibited pyruvate dehydrogenase, so you don't make so much acetyl-CoA this way. And when you're doing this, you're not making any acetyl-CoA because you're not making any pyruvate. So you're really just cutting yourself off here from doing glycolysis effectively from two different points of view. From just the regular point of view and from the, okay, I just stopped glycolysis in its tracks over here with this arsenic stuff. So you're not enabling yourself to use your glucose for energy. So that's obviously bad. Another thing that happens here with arsenic is it stimulates hydrogen peroxide production. Hydrogen peroxide is a reactive oxygen species, and we're going to talk about reactive oxygen species in our next video, but reactive oxygen species are damaging to the cell if you have too many of them. We do have reactive oxygen species happening in the cell as a normal byproduct of metabolism all the time, but if we have too many, we can start to damage the DNA. And here, this picture here, if you notice, this is an old-timey medication bottle here, so you'll notice that people used to put arsenic in the medication. It's chocolate-coated arsenic tablets, like, oh yeah, you get some iron, that's great, but here they put arsenic and strychnine in it. So arsenic, we know what that can do. Strychnine, that's poison too. So good not to live in the early 1900s or the late 1800s when they thought that was a good idea. But this is why that is not a good idea. Okay, so that is it for this video, and next time we'll be talking about reactive oxygen species and superoxides.